Every single cell in your body actually contains about 2 meters of DNA.
Yeah, 2 whole meters.
Which is crazy, right? And it's all packed tightly into a nucleus that is, mere fractions of a millimeter wide.
It really defies logic when you first think about it.
Exactly. Like if you tried to stuff a pair of wired headphones into a pocket with that kind of spatial ratio, you would just pull out this irreversibly knotted mess.
Oh, absolutely. A complete disaster.
But your genome doesn't do that. It's a flawless, 3-dimensional architectural masterpiece. It folds and loops and organizes itself with just astonishing precision.
And that structural precision is entirely what dictates how your cells actually function.
Right.
Yeah, the 3-dimensional topology of the genome basically governs which genes are turned on and, which ones are silenced.
But mapping that architecture computationally, that has turned into, one of the most frustrating bottlenecks in modern bioinformatics.
To put it mildly, yeah, that's a huge headache.
Which brings us to the mission for this deep dive. Today, we are exploring a really groundbreaking paper published in Nature Methods by Liz and colleagues in 2026.
A fantastic paper.
We're looking at these enigmatic 3D structures called topologically associating domains, or TADs for short.
Right, TADs.
And specifically, we're going to dissect the massive computational chaos scientists face when trying to find them. Plus, we'll look at this ingenious dynamic programming algorithm and web tool designed to, solve the problem once and for all.
The overarching narrative here is it's really a fundamental data science problem.
How so?
Well, in biology, we often lack an absolute ground truth. There's no perfect, unassailable answer key hiding somewhere.
Right. Nobody has the ultimate map of the cell to check their work against.
Exactly. So when you have dozens of different computer algorithms analyzing the exact same data set and they give you wildly contradictory results, finding the signal in all that noise is where the real genius of this paper lies.
Yeah. Okay. Before we get into the algorithmic chaos, we should probably establish the physical reality of what these algorithms are even looking for.
Yeah, that makes sense. Let's set the stage.
So if I'm looking at a standard Hi-C contact matrix, which is basically where we measure the frequency of physical interactions between different DNA regions in 3D space.
Right, the Hi-C data.
Yeah, these TADs emerge on the heat map as these really distinct off-diagonal squares.
Exactly. They look like little blocks on the map.
They are essentially localized neighborhoods where a sequence of DNA frequently interacts with itself, but it sharply cuts off interaction with whatever is right next door.
And the biophysics driving those distinct squares is a process known as loop extrusion.
Loop extrusion. Okay, break that down for us.
So you have this molecular motor, right? It's called the cohesin complex.
Okay.
It's primarily made up of these subunits called SMC3 and RAD21. And this motor physically latches onto the chromatin fiber, the DNA.
Like a clamp?
Kind of like an industrial winch, actually. It actively pulls the DNA through its ring, extruding this ever-growing loop.
Okay, wait, let me unpack this. It sounds exactly like pulling the drawstring on a pair of sweatpants.
Huh. That's a pretty good analogy, actually.
All right, you pull the string until the little plastic things at the ends, the aglets get stuck in the hole, and it can't pull anymore.
Yes. And that winch keeps pulling until it hits a physical roadblock. In the genome, those roadblocks are CTCF proteins.
CTCF proteins, got it.
But, and this is a really crucial detail, it's not just about the presence of a CTCF protein sitting there. It's about the geometry.
What do you mean by geometry, like how it's shaped?
More like how it's facing. Those CTCF motifs have to be in what we call a convergent orientation.
So they have to face each other.
Exactly. They must face inward toward the loop. If the motifs are oriented in the same direction, the cohesin motor just slips right past them and keeps pulling.
Oh, wow. So they literally have to be pointing inwards to act as the brakes.
Right. That geometric constraint is vital because it dictates the permanent boundaries of the domain. Once cohesin is halted by those convergent CTCF sites, the loop is anchored.
Okay, but if they're just physical loops, why do we call them functional domains? Like, why do we care about these specific loops?
Because they act as functional insulators. They delimit distinct chromatin states.
So they separate the different neighborhoods.
Exactly. You might have an active domain packed with these H3K36 ME3 marks, meaning there's heavy transcription happening. The genes are on.
Okay, active neighborhood.
Right. And right next door, separated by that boundary, you could have a repressed domain saturated with H3K27E3 marks. That neighborhood is completely silenced.
Ah, I see. So the boundary keeps the active stuff from waking up the sleeping stuff.
Precisely. If you lose that structural boundary, say the CTCF anchor fails, the active regulatory elements from the first domain might bleed over.
and aberrantly activate a repressed gene.
Yes, which is unfortunately a classic driver of oncogenesis or cancer.
Oh, wow. Okay, this is super important then. Let's do a quick checkpoint for you listening at home just to make sure we've got the foundation locked in.
Good idea.
Bullet 1, DNA folds into 3D loops called TDs. Bullet 2, these are formed by a molecular motor called cohesin pulling the DNA until it hits those convergent CTCF roadblocks.
Perfect summary.
And here is a question to ponder for the audience. If TADs are these fundamental physical structures with literal protein anchors, shouldn't it be incredibly easy for a computer to spot them in the data?
You would certainly think so.
Right. But the paper points out that this specific area of computational biology is essentially the Wild West.
The discordance between the algorithms is just staggering.
Let's talk about that experiment. They used a specific cell line, right?
Yeah, they took a highly detailed, deeply sequenced Hi-C dataset from a very well-studied human lymphoblastoid cell line. It's called GM12878.
GM12878, got it. And they bin the maps, right?
Exactly. They bin the contact maps at 10 kilobase and 50 kilobase resolutions. So looking at it through two different magnifying glasses, essentially.
And then they just fed that exact same data into every tool they could find.
Pretty much. They tested 43 different computational calling strategies using 40 distinct software tools developed over the last decade.
Okay, I spent some time looking at the supplementary tables comparing the outputs of these 43 tools, and honestly, they violently disagree on almost every metric.
It's a total mess.
Like some tools confidently state that TADs cover only 50% of the genome. Other tools look at the exact same matrix and claim genome coverage is nearly 100%.
And the sizing is all over the place, too.
Right. The median predicted size hovers around 350 kilobases, but the predictions range from tiny 100 kilobase microdomains up to massive 1 megabase structures.
But the sheer number of predicted domains, that is where the algorithms really fracture.
Yeah, tell me about Armidas.
So the majority of tools identify a few thousand domains across this GM12878 genome, maybe 3,000 or 4,000.
Okay.
However, one tool, Armidas, analyzed the exact same data and called 77,474 distinct TZs.
Wait. Look at this straight. If 43 people look at the exact same map, how can one person see 3,000 cities and another person see 77,000?
It sounds like a hallucination, doesn't it?
Are they hallucinating, or is the 3D genome just inherently fuzzy and fractal?
It's the latter. The consensus in the field leans heavily toward a fractal nested hierarchy.
Like Russian nesting dolls.
Exactly like Russian nesting dolls. The 3D genome is not a simple string of isolated, non-overlapping loops.
Okay, so there are loops within loops.
Yes. You have massive outer loops, what we can call parent TADs. And within the boundaries of those parent domains, the chromatin undergoes further localized, folding into smaller subTADs.
Oh. So they might all be partially right. Depending on the statistical sensitivity of the tool, it might only see the massive outer parent TADs.
Right, it misses the inner dolls.
But a tool like Armidas might be hypersensitive, picking up every single tiny inner subTAD, driving its count all the way up to 77,000.
Exactly. They are all looking at the same structure, just through entirely different analytical lenses.
Okay, checkpoint number 2 for you guys listening. Bullet 1. 43 computational tools were tested on the exact same GM1-202-878 DNA data. Bullet 2. The tools fundamentally disagree on how many TADs exist because the genome folds in these nested Russian doll hierarchies.
Spot on.
Which brings up our second question to ponder. In a world without a map legend or an absolute experimental ground truth, how do scientists play judge? How do they decide which algorithm is telling the truth?
That is the multi-million-dollar question. And the researchers solved it by designing the ultimate algorithmic obstacle course.
Like a decathlon for code.
Exactly. Since they didn't have a master answer key, they had to score the tools based on independent orthogonal biological rules.
So using independent data to verify the computer's guesses.
Right. If a tool predicts a boundary at a specific spot on the DNA, we can check independent GP-seq data to see if the physical proteins are actually sitting there.
Okay, so the researchers broke this benchmarking down into several highly specific metrics. Let's walk through them.
Sure. The first metric is biological enrichment.
Okay, so do the predicted TADs actually do their job, which is isolating chromatin states?
Exactly. A good TAD should be internally consistent. It should be enriched for either those active transcription marks like H3K36Me3 or those repressive marks like H3K27E3.
Right, active or sleeping, not both.
Yeah, if an algorithm predicts a TAD, and that TAD contains a messy, disorganized mix of both active and repressed chromatin, it fails. It didn't find a true functional neighborhood.
But wait, just capturing a shift in histone marks doesn't explicitly prove there is a physical loop there, right? You could just have a linear change in activity without a physical wall.
That is a fantastic point, and that is exactly why the second metric is structural borders.
Checking for the physical walls.
Right. Do the predicted boundaries actually have CTCF, RAD21, and SMC3 proteins sitting there?
The cohesin winch and the roadblocks?
Yes. And they even ran motif analysis to ensure those CTCF binding sites were in that required convergent orientation we talked about earlier.
That is...
Data-driven metrics. So contact enrichment, basically asking, do the genomic bins inside the predicted TAD actually interact with each other more than you'd expect by random chance?
And contact insulation?
Yeah, checking if there is a sharp drop-off in interactions right at the predicted border.
Okay, so we've got biological enrichment, structural borders, and data metrics. Was there a stress test?
Oh, definitely. The final gauntlet is robustness.
How does that work?
Well, high-resolution Hi-C is incredibly expensive to run, so most labs operate with much shallower, lower-quality data.
Right.
So Lai and colleagues took the pristine GM1 TO878 map and systematically deleted the data, downsampling it, until they were left with a mere 0.01% of the original reads.
Wow, deleting 99.99% of the data.
Yeah. And they asked, can the tool still find the same major TADs when the data is overwhelmingly sparse and noisy?
Okay, so I'm looking at the results of this code decathlon, and there's a massive biological trade-off here.
There really is.
The tools that won the histone mark event completely failed the CTCF boundary event. Why can't we just find one tool that wins gold in everything?
It comes back to the Russian dolls. Tools tuned to find tiny subTADs easily pass the histone test, because small regions are almost uniformly active or repressed.
Right, they nail the biological enrichment score.
But because they find tens of thousands of tiny subTADs, they inherently predict tens of thousands of structural boundaries.
And not every minor fold has a giant protein wall holding it together.
Exactly. Many of those predicted boundaries are fake. They lack actual CTCF and cohesin, so the tool fails the structural test.
And the inverse is true for tools tuned to find massive parent TADs, right?
Yep. They excel at structural borders because they only trigger on the strongest, most undeniable CTCF anchors.
But because those parent TADs span millions of base pairs, they encapsulate multiple active and repressed subdomains, making them look messy in the biological enrichment test.
Exactly. Tools like directionality index, arrowhead, OnTAD, spectral TAD, top DOM, MSTD, they all scored at the top of the pack, but they all had these distinct blind spots.
Okay, checkpoint 3, bullet 1, tools were judged on biological markers, structural proteins, and robustness. Bullet 2, every single top tool has a fatal flaw due to this trade-off.
All right.
Which brings us to our third question for you listening. Do we just have to choose between a flawed model A and a flawed model B? Or is there a way to combine them?
And that realization is the turning point of the paper. They stopped looking for a savior algorithm, and instead they built a synthesizer.
Enter ConsensusTAD.
Yes, ConsensusTAD. It's not actually a new TAD caller. It's a dynamic programming concept based on something called weighted interval scheduling. Why battle us?
Bobby, yes. Okay, I really want to break down the mechanics of this because it's brilliant. If I'm following the paper, step 1 is you take the union of all the TADs predicted by the top performing tools. Right?
Exactly. You throw every prediction into one giant pool.
Step 2, assign a consensus score. So if, say, directionality index, arrowhead, and top DOM all independently predict a domain with the exact same boundaries, that interval gets a massive score.
That's the core philosophy right there. It relies on the wisdom of crowds.
Right, because errors tend to be random, but real biology is consistent.
Exactly. False positives are usually random noise. By requiring multiple independent statistical methods to agree, the random noise cancels out, and the true biological signal is exponentially amplified.
Wow. Okay, so what happens in step 3?
The WIS algorithm recursively calculates the mathematically optimal, non-overlapping set of TADs that maximizes that total consensus score.
Wait, hold on. Non-overlapping?
Yes.
But we just spent 10 minutes talking about nested hierarchies, Russian dolls. If it strictly forbids overlapping, how does it handle the subTADs inside the parent TADs?
That is the exact hurdle they had to clear. And they did it with a two-part solution. First, they introduced a flexibility parameter called tmin.
Tmin, what does that do?
It defines the minimum overlap required for two predictions from different tools to be considered agreeing.
Because different algorithms might be a few base pairs off from each other.
Exactly. So they run it iteratively, starting with a strict t-min of 1-point arrow, demanding 100% perfect alignment.
And then they gradually relax it.
Yes, down to 0.85 or lower, to strike a balance between high confidence and covering enough of the genome.
Okay, but that still doesn't solve the Russian doll problem.
But the second part of the solution is a special recursive loop in the code.
Oh.
Once the algorithm finds the optimal non-overlapping set of massive parent TADs, it doesn't throw away the smaller ones.
It looks inside them.
Yes. It takes all the strongly supported subdomains that fit entirely inside those parent boundaries, and it runs the whole WOS process again, just inside that local space.
That is incredibly elegant. It organically builds the fractal structure from the top down.
And in the benchmark data, ConsensusTAD consistently outperformed every single individual tool across the board.
Because mixing 5 flawed recipes actually filter out the noise instead of baking a disaster.
Exactly.
Okay, checkpoint 4. Bullet 1. ConsensusTAD is a dynamic programming framework to synthesize existing tools. Bullet 2. It uses weighted interval scheduling and a recursive loop to find the optimal map.
Beautifully summarized.
Question for the audience. This algorithm is clearly mathematically superior, but if you are a bench biologist without a computer science degree, how on earth do you actually use it?
Right, because an algorithm is only as good as its adoption.
Yeah, if it just gathers dust in an academic journal, what's the point?
The authors knew that, so they built a bridge to the wider community called TADShop.
TADShop, which you can find at tadshop.unil.ch. I checked it out. It's an incredibly intuitive, graphic, user interface web service.
It democratizes the whole process. There are two main features: benchmarking and calling.
Let's start with benchmarking. So if some grad student somewhere makes a brand new tool, the 44th tool, they don't have to manually run all these tests.
Nope. They upload their results to TADShop and it automatically tests them against the 39 existing tools using all the rigorous metrics we just discussed.
That is amazing for standardizing the field. And what about the calling feature?
That's for the everyday researcher. You just upload your raw DNA contact data like a .cool or .hic file, select your genome, and click a button.
And it runs the top tools and a ConsensusTAD simultaneously.
Without a single line of coding required.
Okay, so it's basically like TurboTax for DNA folding. You just plug in your numbers and it does the heavy math behind the scenes.
That's exactly what it is.
Oh, I have a philosophical pushback here.
Let's hear it.
If you make it this easy, just a single button click, doesn't it risk scientists treating it like a black box, like blindly trusting the output without understanding the nested hierarchy or the biology?
It's a fair point. But honestly, lacking a standard way to compare models was holding the whole field back.
Right.
By removing the technical adoption barrier, TADShop actually fosters more robust, reproducible research. It standardizes the playing field so scientists can spend less time coding and more time doing actual biology.
That makes total sense. All right, let's synthesize this whole journey. We went from 2 meters of DNA-forming looped TADs, right?
Yep, pulled by cohesin.
Through the chaotic wilderness of 43 clashing algorithms to the elegant mathematical synthesis of ConsensusTAD, and finally, the accessible hub of TADShop.
It's a massive leap forward for 3D genomics.
I want to leave you, the listener, with a final mind-bending thought that wasn't explicitly answered in the paper.
Oh, the fourth dimension.
Yes. We've spent this entire deep dive treating TADs as static architectural structures, like rooms in a house.
But cells are alive. They're constantly dividing, responding to stress, and differentiating.
Exactly. The DNA loops are breathing, breaking, and rebuilding in real time.
Yeah.
So if these algorithms struggled this much to agree on a single snapshot of a frozen cell line...
How on earth are we going to map the 4D genome?
Right. What happens when the architecture is in motion? That is the next great frontier for bioinformatics.
It's going to be a wild ride.
Well, thank you all for joining us on this deep dive. Keep questioning the structures of life, and we will catch you next time.